now that we figured out how to see those things in 1590 with one of those microscopes, we have a guy, a Dutchman named Anthony Leeuwenhoek, and he was in the 1600s. He was seeing the cells move for the first time. He was really good at making lenses. So that was cool. And then 1665, a little bit after that, a half a century, Robert Hooke first used the term cell, and he came up with it from the term cella, which means small room. In 1830, Matthias Schleiden and Theodor Schwann proposed the unified cell theory, which states that all living things are composed of one or more cells. So this is the cell theory, which states that living things are made of one or more cells. With not having a cell, you're not alive. And so all cells have a few things that they all have in common. And one of those things is a plasma membrane, an outer covering that separates the cell's inside from its surroundings. So that makes a great boundary. So that's a cell membrane, a cell membrane. And that makes an outer covering and makes a great boundary. And then another thing that we have is we have cytoplasm, and cytoplasm is the inside of the cell, and that's sort of like a jelly type like inside. Cytoplasm. So it's jelly-like. How do you spell jelly? This is not the right way. Jelly-like. There you go. And then another thing we have in there, we have instructions for making proteins, because the cell proteins are the things that work in the cell. So we need a lot of those, and so we need instructions for that. And so that instruction is in the genetic material. That is our DNA. The genetic material our DNA are instructions for proteins. And then lastly, the thing that every cell needs to have is machines that are going to make these proteins. And those are known as ribosomes. Ribosomes synthesize or make protein. All right. And then some cells have other organelles that have membranes around them, plasma elementary membranes. And we call those eukaryotic cells. We call those eukaryotic cells. Oops. And then the ones without that membrane, without those membrane-bound organelles, we call those the prokaryotic cells. So those are the this is a prokaryotic cells. Prokaryotic cell. Those have DNA, but they don't have a nucleus inside that hold the DNA, because the nucleus is also a membrane-bound organelle as well. Prokaryotic cells are much smaller than eukaryotes. They measure very tiny. They're like 0.5 to 5 microns. In contrast, we talked about red blood cells a few minutes ago, and those measure about 8 microns, and ovum about 120. Prokaryotes cannot grow larger because they're not having those internal bound organelles limits them, because the elementary membrane lets eukaryotes have much more specialized organelles, and those then can enhance cellular transport of material into the cell, out of the cell, or inside the cell around. Prokaryotes, on the other hand, they rely on molecules just sort of diffusing into the space and then going where they need to go. So they are very limited because of that. In larger cells, the volume becomes too great as compared to its surface area. This makes it more difficult for material to be dispersed automatically, and the cells need these specialized structures helping. The surface to volume ratio decreases. See here on the cell on the left, we have a 1 millimeter to 1 millimeter cell. That gives us 6 millimeters squared as a surface area to 1 millimeter cubed as a volume. That's a 6:1 ratio. When we grow and the cell, let's say, becomes 2 millimeter in size, of course it's very large, but as an example, we have then a 24 millimeter squared surface to an 8 millimeter cubed volume, and that's a 3:1 ratio. So that, in that instance, then is too large, and the distance is too great. And so prokaryotic cells, because of that, are small and they stay small. And if we go larger, we have to get to the eukaryotic state and cell. And so let's talk about the components of a eukaryotic cell. The cell membrane is made of a phospholipid bilayer. Remember from chemistry class, we have a phospholipid bilayer with the polar heads on the outside and then the non-polar tails pointing towards the middle. So that's what makes up the cell membrane. So the cell membrane is a phospholipid bilayer. Made of a phospholipid bilayer. It was first identified in the 1890s, and then by the 1935 it was described as having railroad track appearances. It was also compared to a sandwich, which makes sense. The hydrophobic tails can be the meat in the middle of it. And then in the 1950s, microscopy advances made it possible to see the double layer. And then in 1972, we introduced the fluid mosaic model. Fluid mosaic model. Here we go. Fluid mosaic model. Oops. And that is a very nice model that lets us visualize how the different components or mosaics fold around and change positions within the membrane without disrupting its entire integrity. It does that because of the inward pointing tails on the phospholipids, creates that flexible oil film. You see that in here, these tails create that flexible oil film that sort of suspends as a whole unit in that liquid environment. This oil film then makes a clear boundary between the inside and the outside of the cell, which allows both the inside and the outside be their own very distinct environments. Fences, I guess, make good neighbors after all. It's a law of nature, so to speak. Only water-repelling, hydrophobic or lipid-based molecules can cross through this cell membrane and go in, or out or in. But water-based or hydrophilics, they are not allowed to do that. So they are not able to do that. So they will be crossed out right here. The cell membrane is also known as the plasma membrane. So cell membrane is also known as plasma membrane. Well, actually it's two words. Plasma membrane. It's also known as the plasma membrane, or even as the elementary membrane, because we understand its fundamentalness at this point. As I mentioned, the cell membrane is very flexible. That fluidity is partially established by the unsaturated fatty acids, such as omega-3 we mentioned in the chemistry class. When the membrane's fatty acid chains contain them, they're kinks. create these double bonds and make them pack loosely, and that looseness creates that fluidity. In comparison, if we take saturated fatty acids, they pack very tightly because the single bonds rotate easily, and unfortunately they can just pack very tight one next to the other. That makes it rigid, and this can make cell membranes susceptible to rupture, especially when we have a cold environment, and it makes also harder for molecules to cross it. We can see that right in here. Where these are saturated, this is a cell membrane, it's a little hard to see. These are long, straight lines, these are saturated fats. This molecule cannot go through it easily and penetrate it. On the other hand, here you see the kinks in it, this is much more loose, there's space in here. This is an area where the molecules can cross much, much freer. Another interesting implication I remember from chiropractic school when Dr. Bruce Lipton from Stanford came and talked to us about the importance of the cell membrane in cellular communication as compared to the nucleus, which of course gets a lot of attention because it's so sacred as holding the instruction booklets for protein. But the cell membrane, the outside of the cell, is where we communicate. In that discussion, the importance of the membrane fluidity not being too rigid came up. Particularly when we see measurable differences between people now and many years ago of these different structures changing. That is some food for thought. And I think that is a fundamental reason why limiting consumption of fatty acids or saturated fats, and especially trying to avoid the trans fats, which are so often in these uber or over-processed foods, becomes very, very important, I think, and it's definitely something to think about. All right. Cholesterol also greatly assists the balancing effect temperature has on the cell membrane's fluidity. It's kind of like a buffer. When it's too cold, it makes it more fluid. When it's too warm, it stiffens it up a little bit. It also helps organize clusters of transmembrane proteins into lipid rafts. Very cool stuff. Proteins are embedded in the cell membrane as well and attached to the surfaces also. They have multiple different functions. Where are the proteins? We got integral proteins right in here, and then we got peripheral proteins that either attach to the inside or the outside of the cell membrane. Some of them are channels that create passageways for molecules like glucose to go in and out of the cell. We have one that looks a little bit like a channel like here. If that changes shape, it can potentially open and something can go in and out of the cell. Other functions like enzymes, which help promote specific chemical reactions. Even other ones recognize messaging proteins like hormones, and then with them attached, those hormones change shape, and then that initiates a reaction on the inside of the cell, such as making specific proteins that are necessary or so. Some other proteins are involved in connecting the cells to one another. Like, for example, if you pull on your skin, those cells, if they weren't strong enough, your skin will rip apart, but it doesn't because they have strong connections between them, and those are also proteins. We'll talk about those in details a little bit later. And then lastly, we consider, for example, the inside space of a cell. To maintain its volume, it needs to have structural support, which many of the filaments and microtubules carrying out. And they're often also attached to the cell membrane. We'll talk about those in a little bit as well. Carbohydrates attached to proteins or lipids on the outside. See here, glycoprotein. You see here carbohydrate, and then you see here one that's attached to a lipid directly as well. And they attach to them, and this then creates on the outside some sugary film, and we call that sugary film the glycocalyx. See these fuzzy things sticking up? Those are those carbohydrates. So that is called the glycocalyx. Glycocalyx. Oops. There you go. It is unique to each individual organism, and it helps us, and then helps our immune system to recognize ourselves. And then if another cell doesn't look like the same, the immune system can recognize it as foreign and then try to destroy it. It's amazing how all of this works together, and how shape is such an important fundamental way of how our body works. It's really like the lock and key kind of mechanism, how things work together and how recognition of these shapes does so many things in the cell. And of course, this brilliant lock and key mechanism is also vulnerable and can be manipulated, which is what viruses do. Since they can't do some cell functions, they find a host and then go into it. They mimic the shape of the binding sites of a cell and enter the cell, and then they go into the cell and manipulate it and do the tasks that they cannot do themselves. The membrane of cells is involved in absorption as well. So let's talk about that next.